So today we've had some great presentation on exoplanets and other galaxies, and so now I kind of want to switch gears a little bit and go to the teeny tiny scale, or micro scale in our oceans. So in the next couple minutes, I'm going to give you an introduction to my research, what I've been doing more or less 40 hours a week while also going to school at the University of California. My project, some of the cool things I think we found, and wrap it up with some of our conclusions. So I study the influence of engineered nanoparticles on phytoplankton. Engineered nanoparticles are man-made particles with diameters ranging from 1 to 100 nanometers. They're applied in medicinal therapies and industry. They're really common in sunscreens and cosmetics, as common as they are on photovoltaic cells for solar cell technology. Their applications appear limitless. There's more and more nanoparticles added to things every year, and to that end, their environmental impacts are relatively unknown, which is worrisome. So a general model, marine phytoplankton are tiny microscopic plants that float on the surface of our ocean. They serve as the base of the ocean food web. A major portion, around half of fixed carbon of these algae is released as EPS. This EPS, or exopolymeric substances, think of long chains of polymers, assemble. And this assembly process in the presence of this nanopollution is what I study. So basic model, we have the sun. The sun feeds the algae on the bottom, your bottom left-hand corner. Through photosynthesis, they excrete EPS, which contributes to the DOC pool, dissolved organic carbon. We mentioned that they spontaneously assemble, and this assembly process is important for marine gels, a means of carbon sequestration, also a means of food for high organisms like zooplankton. So we studied the way that these polymers assemble with time. The two things to note are the bottom red, which is the control groups for 3 phytoplankton species, and the top, which are in blue and black, which are 10 and 100 parts per billion concentrations of engineered nanoparticles. The engineered nanoparticles that we used were nanopolystyrene. Notice that the y-axis is the hydrodynamic diameter, so when EPS assemble in the presence of nanoparticles, again, in really low concentrations, parts per billion, they have really large effects. They not only accelerate at which they assemble, but also have a larger diameter size once they plateau. This is sort of looking at kinetics. We wanted to know also what these look like. So we took scanning electron microscopy images. The images to your left are consistent with the dynamic laser scattering measurements, in that they have a larger radius. To the right is the control. You see sort of scattered particles coming together. And we also used epifluorescent microscopy to look at, we've established that the nanoparticles not only make the gels larger, but we wondered, because the gels are eaten by higher organisms, are the gels incorporating engineered nanoparticles into their gel matrix, which might raise questions for potential for bioaccumulation. Things eat these gels, which things eat those things that eat these gels, and so on up the level. So we wanted to find a way to say, okay, how do we tell whether or not engineered nanoparticles are being incorporated into the gel matrix? So we used epifluorescent microscopy to do that. On the top row are all pictures of gels using these EPS gels using epifluorescent microscopy. And then important to note is, with the engineered nanoparticles, which are in green, we wanted to see if the gels had nanoparticles in their matrix. Then when we looked for those gels with epifluorescent microscopy, we would see not only the red gels, but also the green gels. So because the green gels are missing in our controls with no nanoparticles, it lets us know that there are nanoparticles in the gels with 100 parts per billion and 10 parts per billion concentrations. And this work was published in Public Library of Science Journal 1, a paper of which I'm the co-first author. So to kind of wrap up, we found that engineered nanoparticles accelerate EPS growth and increase their microgel diameter, which is very interesting in terms of these microgels stay on the surface, and if they get larger and they sink, it could be a way of carbon sequestration in the ocean. They might also take with them nutrients essential for phytoplankton growth. So if it takes with them, then there's less nutrients on the surface for phytoplankton to grow, these algae. Moreover, more worrisome for bioaccumulation risks, their engineered nanoparticles can be incorporated into the microbial matrix. And finally, sort of on my future goals, I'm also interested in quantifying other forms of marine pollution. So we looked at nanopolystyrene, but it's one of many engineered nanoparticles, and so we want to see sort of if we see the similar effect in other nanoparticles, as well as more than 3 phytoplankton species. And I've been doing a lot of science stuff, and I'm really interested in the policy implications, so that's kind of where I want to work from there. Thank you.